So, good evening to everyone. Welcome to the webinar of the Biological Physics Commission of the Brazilian Physical Society. Today we have a researcher from Chile, Professor Ignacio Antonio Bordeu. Thank you very much, Professor Ignacio, for accepting our invitation to present your webinar in our section of the Brazilian Physical Society. So can you please share your screen with us and start your talk, please? Welcome and thank you very much.
Thank you, Marcio, for the introduction and for the invitation. Just to make sure, can you see my presentation now?
Yes, it's
Yeah.
okay.
So it's a pleasure to be here talking to the Brazilian Society and especially to the biophysics community there. Today I'm going to talk about branching morphogenesis. So branching morphogenesis is a developmental process that gives rise to branching organs and structures in our bodies. And I'm going to talk about it from two perspectives. One is the more physical side, and also I'm very interested in answering real biological questions about these processes. So you're going to be seeing a lot of things like what you're seeing now in the screen. So spatial branching processes and how we can connect this to biology. So as I was saying, branching morphogenesis is the developmental process that gives rise to these branched structures that you can see mostly. You can imagine the lung, and the lung is a tree-like structure that develops from a single duct, and that duct divides into 2, 4, 8, 16 branches until it forms a fully developed tree. You can see those structures in the kidneys, pancreas, mammary gland, salivary gland, lung, vasculature. So any tissue that transports fluids in our bodies has adapted to form a ramified structure because it optimizes space and it optimizes fluid flow. In general, depending on the tissue, people have discussed that there's different modes of branching from the idea that there's side branching. So you can imagine a single tip that is elongating. So you can, in every tip, you're going to have stem cells, and those are cells that divide rapidly. So they're actively proliferating. And as they proliferate, they're gonna elongate the ducts, and there's different modes of branching depending on the tissues. In the mouse lung, people have argued that there's different modes. Some of them are planar. There's also some modes that are correlated. So you can see that there's alternated planes of division as the tissue develops. Others have argued that that's only because of mechanical constraints in the tissue, and it's not a real mode of branching. It's just due to mechanical constraints. But in general, these processes involve a multi-scale range of processes from the cellular level, even the molecular level. You can imagine a single tip, as a collection, as a multi-layered epithelium that has stem cells, has differentiated cells, and there's duplication and there's transport of cells to the ducts. And as this process happens, the tips elongate. This has to live with luminal pressure. So you can imagine that as these tissues develop, they are forming a lumen. A lumen is a cavity, is the inside of the ducts that may be filled with fluid, and there might be peristaltic movements there. So there's fluid that might be changing in pressure as the tissue develops. There's, of course, a surface tension type of mechanism due to the addition between cells. There's accumulation of extracellular matrix components. So you have to imagine that this tissue that is it's an epithelium is surrounded by a scaffold. So a second type of tissue that is a mesenchyme that is a scaffolding, but it also interacts with this epithelium. So there's a multi time scale and spatial scale process going on. And there's, of course, some reaction diffusion interactions between morphogenes that are exchanged between the mesenchyme and the epithelium. So I say all of this to convince you that this is not a simple problem. The idea that tissues are branching is not a simple process. And I'm not going to try and answer all the questions that you might have. I'm going to try and connect what we have observed in different tissues with the large-scale organization of these tissues. So I'm going to try and argue that the large-scale organization, so these trees with multiple branches, develop in a way that is self-organized, and it's not necessarily embedded in our DNA the way these organs develop. So to go to the large-scale organization, I'm going to focus on some work that was done some time ago by Hanesso from the group of Ben Simons in the UK. And what they try to do is to simplify the process of ductal growth and elongation as a sort of picture of interacting particles. So let's say that we have an elongating tip, and that tip is a particle. And that tip can deposit, well, it can move, first of all, so there's elongation. Yeah, so the tip can displace itself to a new location, and you can imagine this as a persistent random walk. So the particle is moving. And as this particle moves, it's going to deposit immobile particles that are going to end up forming the ducts, right? So these are going to be immobile particles. And at the same time, as these tips proliferate, the tips are going to be able to branch. Yeah, so an active particle is going to be able to produce two active particles. But because these ducts have a volume and there's a volume exclusion process, so there's steric interactions between the ducts in the tissues, and that's mostly in the organs that I mentioned before. But in the vasculature, for example, you can see that there's reconnections. So sometimes the steric constraints are not so relevant. In particular in the vasculature, that are not so relevant because there's reconnections. In all other tissues that I mentioned, reconnections are rare or don't exist at all. Yeah. So what's going to happen is that when a tip that has a lot of proliferative cells approaches another duct, the cells in this tip are going to exit cell cycle collectively, and they're going to arrest the growth of this tip. So they're going to become inactive and just fall out of cycle. Yeah. So these were the rules that were incorporated by Edward Hanesso and Ben Simons and company some years ago, quite a few years ago, 2017, to understand the growth of the mammary gland. So what's good about the mammary gland is that its ductal structure seems to be quite homogeneous. So there's no huge ducts in diameter compared to others. So the tissue seems quite homogeneous. It's also a 2D structure. So it develops in the pubertal stage of the mouse, and it develops on a flat surface. So it's easy to image. It's easy to segment, fairly easy to segment. And another thing that it makes our lives easier from a modeling perspective is that it grows on a fixed domain. So when the ductal structure is growing, and you can see it here, these are 3 different samples. There is a yellow outline here that's going to be the domain. So you can imagine if you have a more numerical simulations kind of mindset, you can imagine the yellow outline to be your simulation domain, and that's fixed throughout your simulation. And what's going on is that the ductal network is developing from this point outwards. So it starts from a single tip that forms multiple tips as it's developing. Okay. And so using this simple rules that I mentioned before, so elongation, branching, and inhibition, they thought, well, maybe we can use these ideas to model this branching organ. And what they did was to analyze the data. They realized, for example, that if they looked at the ducts at different generations of branching, so this is the root here, the origin is what I showed up here as the origin. Yeah, so it's the beginning of the network. You can hang the tree from that position and look at the topology of this tree. You can look at the widths of the ducts as a function of the generation, and that's what's being shown here. And you can see that it's not changing, so it's fairly homogeneous. You can also see that if you look at the distribution of duct lengths, you're going to see that distribution falls pretty much on an exponential decay. This is a log linear plot. So this means that you can interpret the branching process as being a Poissonian process. So imagine a tip that is elongating at a fixed speed. If the timing between events is exponentially distributed, then the length of the ducts are also going to be exponentially distributed. So this is fairly, it's pointing towards a Poisson process of divisions. So there's probably no memory in these branching events. So this makes our lives easier because we can use a sort of a rate process, a reaction diffusion process to model this. And that's exactly what they did. So they said, well, branching events are uncorrelated. They occur at a constant rate. Each tip describes a persistent random walk. And active tips are going to leave a trail of inactive ducts. Then they can inhibit the active tips. So I'm just going to summarize a bit. So remember the mammary gland. Remember that the process is starting from a single tip. And the only things that are happening here are elongation, tip branching, and some sort of inhibition. Yeah, and we can discuss what's the origin of this inhibition. If it's purely steric, if it's mechanic, if it's mechanochemical, if there's a gradient of interaction here. But that's for another discussion. Yeah. So what they realized is that if they simulated this process, what they saw is that there is an elongation process, and this is being simulated on a rectangular box. Yeah, the box is not here, but you can imagine this is in a box. So what's going on is that all red tips are active, so they're actively moving and proliferating and depositing duct. And what happens is that you can see immediately that whatever is submerged is inactive. So it became differentiated and just fell out of cycle. This is a numerical simulation, by the way. So what's going on is that the tips that are active are those that remain at the periphery. And you can see that here, all of these have touched the boundaries, but the only ones that remain active are those at the periphery. So this is already a self-organized quality of this process, right? Because of the inhibition, we should see that proliferation is mostly at the periphery of the tissue. Of course, because of the way the model is constructed, you immediately see an exponential decay of duct length, because that's by construction. So we're assuming a Poisson process of duct of bifurcations. So, but one thing they did was to actually measure this proliferation in the tissue, in the actual tissue. And they did this by EDU, which is a proliferation marker that is incorporated to the cells during cell cycle. And you can see immediately in this ductal network that hasn't developed fully, is still in development, that all actively proliferating tips are those at the periphery. So this is elongating and branching in this direction into the empty space. But everything that is left behind has already deactivated, became inactive. Right, so it's these are good indications that the model is fairly close to what's going on in reality, or at least it models in a good way what's going on in reality. Another thing that you can do is try to do some maths with this. You can approximate this model mean field by well, zooming out, and you look at regions of the tissue and you ask, well, what's the probability of seeing a branching event? Well, that's going to be effectively it's going to be an effective rate of branching with which particles are gonna divide into two particles. Remember, this is all local, so this is occurring at a single point in space, but we're looking at mean field, so very far away. We're going to have a deposition of particles as well. So we're going to say that A are active particles. Those are going to be diffusive in our mean field equations. We're going to have also inactive particles. Those are ducts that are being deposited by active particles as they elongate. Yeah. And if an active particle interacts with an inactive particle, then they're gonna become inactive. Yeah. So it's gonna be just an inactive particle. Yeah. And the same happens if two active particles meet. But of course, this occurs with very low probability. What's happening all the time is interaction between active particles and ducts. Yeah. And of course, you can build the mean field, the rate equations for this, assuming that only active particles are those that move. So we're gonna, at this level, we're gonna say that these particles move diffusively. You could make it more complicated and say, well, let's have an actual active Brownian motion here, but for the sake of this analysis, it's not quite relevant. And what happens is that the density of active particles forms a pulse. So you can see what's going on here. These are different time points. And you can see that active particles form a pulse that organizes at the periphery. So you can imagine here a density as a function of the x-axis. So there's no density of active particles and suddenly a peak that decays to 0 again. Yeah, so that's a pulse of active particles. And what's going on with inactive particles is that it's being left behind by the pulse of active particles. So there's a constant density of duct in the wake of this wave that is propagating, yeah, this pulse that is propagating. And this is described by this mean field model. And if you go to the tissue and analyze what's going on between tips and ductal density, you see that the model captures quite well what's going on in the tissue. So this is all of this is to try to convince you that this is a good approximation to what's going on in a family of branching tissues in our bodies and in the mouse and in different animals. Yeah. So I want to do a break here and talk a bit about the statistical mechanics side of things, because this model had been studied before in the 80s by, well, Cardy, Grasberger, Janssen. These are big names of the field, right? And the reaction diffusion process they had studied was just exactly the same as we had before, without the tip inhibition, but that's not really relevant because it happens with low probability. And what's nice here is that they call this model a general epidemic model. So you can imagine this as being an epidemic that is propagating on susceptible individuals, which is the lattice, the full lattice. And as individuals are being infected, they cannot get infected again. So when the infection reaches them a second time, the infection just dies off, right? And you end up with a process, well, the dynamics is quite fun. Again, of course, you see this ductal network formed, and you see this front of active particles propagating. And what has been shown, and we've continued studying this from a statistical field theory perspective, is that, well, this model first, because of its interactions and the branching in particular, it has an upper critical dimension that is 6. So it's actually mean field works from 7 and above. But because we're in the supercritical regime, so our processes always explode, we're safe on doing mean field in lower dimensions. But what's interesting is that, well, the growth process falls in the universality class of dynamic isotropic percolation, and in particular, the pattern that is left behind. So what you see at the very end here, now can I stop this? Yeah. What you see at the very end, that pattern has the properties of of standard percolation. So you can use results of standard percolation to understand some properties of the final state of this system. You can see that because the process has these steric interactions, it leaves behind holes, and it has some density fluctuations that are interesting to study. In particular, we've been studying those with Marius Bothe. He was a PhD student at Imperial, and he's now working in a quantum computing company. He left us, but he's still quite engaged with the project. And so he's been doing the statistical field theory calculations and reproducing some of Cardy's and Janssen's results. And in particular, we've been interested in understanding how these density fluctuations vary with the branching rate. So what are the scaling exponents of density fluctuations? And by density fluctuations, I mean, if you take a box of size L, or you divide your system in boxes of size L, and you measure what's the number of particles inside of the box, of each box. You calculate the mean of those number of particles, and then you take the variance of the number of particles, you can plot those in a log-log scale. And in the, let's say in the mean field, you would expect, in this case, we expect an exponent of 4. But we're standing at the critical point in the calculations and looking at those corrections, right? We're just doing an epsilon expansion, so we're looking at d equals 6 minus epsilon. Yeah, so we're studying those density fluctuations, and in particular, I'm interested in understanding what's the meaning for the biology of those fluctuations. There's another thing that you can study, and this were drawn by myself by hand. You can look at the holes. So what's the distribution of the hole sizes in these cases? And those are interesting because those are regions that are sort of unoccupied by the biology, by the tissue. And one would imagine that that's a bit inefficient, right? And it turns out that some tissues are much more efficient at filling space than others. And the question is, in the other tissues, how does the biology fill the gaps? Yeah. So in some tissues, there's side branching. So there's this idea that also ducts can reactivate and fill the spaces that are empty, that are free. And that requires a reprogramming of the cells. The ductal cells have to become more stem again and produce these new ducts. And there's, of course, from the percolation community, there's exponents measured for this distribution of holes, which is interesting. So okay, let me stop with the more stat-makey things, and let's go back to biology now to one of my projects. So I want to show you some results that we published last year. And this is going to be on a different tissue. So at the beginning, I talked about the mammary gland. That's a 2D tissue. And what we're going to do now is we're going to go to a slightly more complicated tissue. Yeah. And that's the mouse salivary gland. So it might not sound very exciting. It's just producing saliva, right? But it turns out that it has many features that the mouse mammary gland doesn't have. In particular, the mouse salivary gland, it develops during development, so during embryonic development. So the salivary gland, sorry, is expanding with the embryo. So it's growing within the tissue that is expanding, right? We're gonna talk about that in a moment. And the other thing that is interesting is that the salivary gland is a 3D object already. Yeah, so you don't only have a 2D lattice that is being filled, you have now a 3D volume that you would like to occupy. Yeah. So this gland that we're going to focus on is a submandibular one. It's around here, and it develops within around 7 days. It starts at embryonic day 11.5, with this lamination of a plaque coat that then forms a single duct, and this duct bifurcates in series until it forms around, well, of the order of 1,000 ducts. Yeah, around that's the order of ducts that are being formed. Yeah, and it has multiple cell types. It has ductal cells that are luminal, basal, and then there's the acinar cells that are produced after the tip cells differentiate. Those are the ones that produce saliva, and the saliva is pushed out from the tips by with help of these myoepithelial cells. So these are contractile cells at the periphery of the acinus. So the acinas are in all tips of the tissue. They produce saliva, they squeeze it out, and the saliva is pushed through the ducts out through the main duct and into our mouths. So it's a very nice process of fluid transport there as well. So there's a few questions from the biological side that we as quantitative people can help answer. And this is something we did with Lemonia Schatzelli. She's now a postdoc in Ben Simon's group. And she wanted to understand how the lineages develop and how these tip cells, that are the ones that are actively proliferating, form all the tissue, all the full tissue, right? So they're producing this luminal, ductal, acinar cells, and so on. I'm not going to talk a lot about that, a lot about this, but if there's any questions, please ask me whatever you would like to know. So what we did was to segment one of the lobes of the salivary gland. This is a single lobe The salivary gland is quite bigger than this, but this is already hard to quantify. So this has around 2,000 ducts, and you can see it moving around here. So the segmentation of the ducts was done manually. We tried machine learning, but it didn't quite work for this setup. We're now using it for human salivary gland, and it's working much better. So this one was done manually. Lemonia traced all the ducts. And then what we did was to label individual cells at the beginning of development near 13.5, so quite early on up here. We labeled individual cells. It was more around this stage where we have more tips. And then we look at what's the output of those cells. So we look at that much later in development. And we see these clusters of cells labeled from a single color. So we know that here what you see as dots, color dots, are single cells that are labeled. And those that share the same color are all descendants of a single cell that was labeled quite early in development. So this gives us information of how the tissue is producing the different cell types of the organ. So we can then go back and see what types of cells are each individual cell here, and we can build a whole hierarchy. And we can build a whole hierarchy based on our observations. So we can say, well, there's a multi-potent NBAT precursor, and due to some expression of some, in some cases notch, in other cases is K14. These are all molecules that are being expressed by specific cells. You can end up specifying lineages of cells. So this was done with Lemonia, and it was a nice publication that we got out of this. This was all doing quantitative analysis, but then we wanted to understand the actual tree. So how does the large-scale organization develops? So that's what I'm going to talk about now, which is more into the physics of things. So for this, we don't really need those labeled cells. The only thing we need for the moment is this tree, right? So we need the branching structure. And hopefully we need it, and actually we need it at different stages. So we did this segmentations at 3 different stages for 9 different mouse. So we got quite a bit of data on the topology of these trees, the properties of the ducts, the lengths of the ducts, and how they develop over time. And the first thing we realized in this tissue, as I was saying early, is that there's different stages of growth. The first thing we realized is that regardless of the mouse that we looked at, there was always a few levels of the tree where we saw no terminations. So you can see this is just a topological tree. There's no duct lengths here. So each generation corresponds to a branching event or a termination. And terminations begin only after 6 generations of branching. So we have 2 to the 6 ducts here, and only then we start seeing the first terminations. So we call this early stage a stereotypic phase because there's no terminations, and all that is happening is branching events. We can understand them as binary branching events. And that happens because we have, well, we believe, and I'm studying this at the moment, this happens because we have different sizes of tips. So early on, the tips are quite big. They hold hundreds of cells, and they can divide. They can have multiple divisions in parallel. So they can divide. It's like a budding phase, right? You have a single tip that makes multiple divisions quite fast. But later on, at embryonic day 15, 16, and 18, what starts going on is that you have only binary branching. So now we have this tip bifurcation event, only bifurcations, not trifurcations, not more than that. Yeah. So what we're gonna try to understand is the later stages. So the stages that are non-stereotypic, those that apparently follow a stochastic rule of branching. And as I was saying earlier, one thing we realized quite early on is that this tissue inflates over time. So you can imagine that the embryo is growing during development. The cavity where the salivary gland is located is expanding, and the salivary gland itself is expanding over time. And it's not expanding linearly, it's expanding exponentially. This was a quite striking finding, because it made us question the things that were assumed in the earlier model for the mammary gland. If the tissue is expanding exponentially, what must be happening is that all cells in the tissue must continue proliferating. Because if only cells in the periphery proliferate, then you would have a linear expansion with time. So what must be happening is that all cells in the tissue must continue expanding, so that every 2 points in the tissue must expand exponentially. So this is like the inflation of the universe, right? If you take any 2 points, they are getting further and further away exponentially over time. So the first thing we did was to study proliferation, and we immediately saw that proliferation was everywhere. So this is a cross-section around the center of the tissue. And what you see here in red is regions of the tissues that are proliferating. So we saw proliferation everywhere, not only in the periphery, as in the mammary gland. This was a good indication, and it was consistent with the idea of exponential inflation. And at the same time, we saw that every quantification that we did over time, so at 14.5, 16, and 18.5, they all changed with time, not like in the mammary gland, where measurements tended to be stationary in different developmental points. In particular, we saw that the distribution of that length that earlier I showed, it was exponential, here acquired this long tail with time, consistent with the idea that older ducts have more time to elongate because they have been submerged in this inflating tissue for longer. Yeah. So so this was striking, and we had to somehow incorporate these ideas into our model. That was still this idea of branching and elongation, but now we had tissue inflation. So we had this expansion of the tissue. Yeah. So what we did was just that. So what I did was to take this this idea and say, well, let's assume that whatever is left behind, actually every particle in the system, is standing on a tissue, on a 3D tissue that is inflating exponentially over time. So this means that the network itself is getting bigger over time, but this has implications on the annihilation, on the inhibition process. So I was saying earlier that when a tip comes close to ducts, there is an inhibition process, and this implies that a tip is going to become inactive. But if the tissue is expanding, then new space can be opened between this otherwise inactive tip and the network. So you can imagine that there is a tip that approaches a duct, but the ducts at the same time are separating because there's cells in between that are proliferating. And this means that these otherwise inactive tips can reactivate and continue their branching process. Yeah, and this is going to change. This is going to change the network, because it's going to continue elongating but also branching. Right? It's going to contribute with further development of the network. And this is how we came up with this idea of an inflationary branching and delayed random walk. The idea that there's a branching random walk that is occurring on an inflationary background, and that causes submerged tips not to be annihilated, but to be delayed. Yeah, so it's a bit of a change in the dynamics. And if we think of these reactions that I was showing earlier, so now we have tips that can bifurcate. This is the same as earlier. We have tips that deposit inactive ducts. This is the same as earlier. We have these inhibition processes, which is the same as earlier. The only difference now is that you have a new letter here. So what's this new letter? The idea now is that we have active tips. Those are the ones that are actively proliferating. We have inactive tips. Those are the ones that are being deposited as ducts. And we also have delayed tips. So these are tips that are restricted by the tissue. Yeah. So when there's inactivation due to interactions, tips are being delayed. They're not deactivated fully, and those can reactivate. So delayed tips can reactivate into active tips at the rate of the expansion of the tissue. Yeah. And this is actually like this. So if you expand exponentially, then the rate at which they should reactivate is proportional to the expansion rate. So we have two things due to expansion. One is this reactivation, and the other one is that ducts need to elongate. So whenever you have a duct, you need to form two new particles so that the tissue remains connected. Yeah. So there's an increase in the number of ductile particles, and you can build a mean field model out of this. It looks messy, but it's actually quite simple. It's just taking these equations and writing it down. We have diffusion of active particles. We have the branching rate of active particles, the inhibition term, which is nonlinear, and the reactivation of delayed tips. So we have now three equations, one for active, one for delayed, and one for ducts. Yeah. And the only thing that is quite new is this term here, where we have an advection and dilution term. So you can separate this into two terms, and you're gonna realize that one is an advection term due to the inflation of the tissue, and the other one is a dilution term. That is, as you inflate the tissue, the density of particles is being reduced by this inflation. Yeah, and that has to be compensated by branching and elongation and so on. Yeah, here U is just your inflation field. So we're assuming that the inflation of the tissue is causing some sort of flow in the particles. Yeah, it's an advection of particles due to the inflation of the tissue. And it turns out that if this flow is causing an exponential inflation, so it's proportional to the coordinates of the system, then this is quite easily expressed just in terms of the rates, and you can do some rescalings and actual calculations with this. So what can be done here is that you can calculate the stationary states. So you can find what are the stationary states in terms of on the rates of the system. And you can see here, this is the stationary state of active particles. So you can see that then when there's no expansion, so imagine that the expansion rate is 0, the only stationary state is active particles equals 0. So you have a field of dead. The system is dead. You cannot have active particles if your system is fully covered by a network, because all tips have exited cell cycle due to these steric interactions. And in that case, in the case without expansion, you can have only a field of inactive, a uniform field of inactive particles, right? In this case, because we have expansion, we have a condition for this stationary states to exist, and it's that there must be a balance between the branching rate and the volumetric expansion. So this is the expansion rate in one dimension, but you have to multiply it by the 3 dimensions, and that's the volumetric expansion. And as long as the branching rate is large enough, there's gonna be an active stationary state where active particles are continuously producing new particles to fill the space that is being opened by inflation. And what you end up with, well, here I'm showing just a solution of the system of equations I showed earlier, starting with a single particle here at 0. This is space, and you can see that there's a pulse, but now it's an actual front, right, of active particles. It's this one. It's a front that is propagating in the direction from left to right. And as it propagates, it left behind a field of active particles, delayed particles, and ducts. And these are all being normalized by the stationary state. So what's being left behind is a mix of all particles. Yeah. We compare this with the actual tissue. So by looking at these images like the one I showed earlier, you can analyze the density of proliferation as a function of the distance from the edge. So from the yellow periphery here. And we saw an increase in proliferative activity in the epithelium near the periphery. So 0 is the periphery, and it decayed towards a more constant value away from the periphery. while the mesenchyme, which is the submerged, the scaffolding, let's say, that is constant overall. Yeah, so this is consistent with the idea of having a peak of active particles in the periphery, but a homogeneous state in bulk. Yeah. So you can go ahead and do your particle, your agent-based model as well. So there's the measurements that you can extract from the tissue, branching angles, all sorts of things that you can measure from the actual experiments and use them as inputs for a particle-based model. Yeah. And we ended up with a model that is, it's basically parameter free because everything is being inputted from the experiments. And then what we did was to just simulate this process as a simulation that started from our earliest time point that we had data for. Yeah, so this is it begins with the stereotypic tree, and then we simulate the non-stereotypic phase of growth. And you can see here on the very right the actual branching tree as it develops and some topological quantity that we measure. I can explain it later if it's necessary. So we're simulating here 84 hours of growth. Yeah, and we can then match those results to the actual experiments, both the topology and morphology, and we see a good fit. I have some plots later on. So one thing that our model predicts is that even though the tissue has steric constraints, submerged tips still have the potential of branching, right? So one thing that our model predicts is a lower but persistent branching process in bulk. So we should be able to see this in experiments. We cannot see it in vivo because we have no way of reaching the salivary gland of the mouse in vivo. All the images I showed earlier are from salivary glands that are extracted from the mouse and then imaged. So it's a dead tissue. But what we did was to do an explant. So we took out a salivary gland of one of the mouse, and Lemonia managed to image this ex vivo. So this is in a petri dish. And what you can see is how the tissue evolves during 40 hours. Yeah, and we imaged every hour. So what you see there is this drastic inflation of the tissue within 40 hours. And what we did next was to quantify some trees that were near the periphery. And what we found was that if we focused on a tree, this is early on, this is the same tree that is being tracked down, and we focus on tips that are near the periphery. So those are the blue tips here that are labeled down here. And we look at them 40 hours later, we see that they develop into these large trees with many branching events. But if we focus on tips that are submerged, those are the black ones here, and we follow them for 40 hours, we see that they still branch, but much less than those that are at the periphery. This is not statistical. This is just an example, right? It's difficult to do this analysis, and that's why we did it for a single tree. But it's still consistent with the predictions of the model. Yeah. And as I was saying earlier, there's many quantifications that you can do at different time points from the particle-based model. So from this, the actual branching structure, to show that the model actually predicts all of the features of 2 time points without fitting parameters, basically, right? So it points in the right direction. It's telling us something that all of the complexity of these branching tissues is not necessarily encoded in the DNA, but it's self-organized, right? Even though in some of these tissues there's coordination, there's local coordination, or there's global guidance mechanisms, and you can imagine tissue inflation as being a guidance mechanism as well, like an external potential somehow. It's still self-organized. The tissue is responding to local cues in the environment and reacting to them in order to fill space efficiently and fulfill a function. Yeah. So with this, I want to finish. I just want to acknowledge quite a bunch of people. So of course, I want to acknowledge Ben Simons I was in his group, and I'm continuing working on this project. I have now new avenues and branching events also in physics, and that's why I put some people from the University of Chile here. We've been studying branching events on liquid crystals as well. With Lemonia, we're now exploring the human salivary gland. With Ivan here and Megumi and Alen and also Ben, we're studying the human lung, and it seems to have different rules of branching, but it's still a stochastic rule seems to work well in the human lung, even though it's a much more structured tissue and has a fractal structure. I also want to acknowledge the work done by Marius and the people, my collaborators at Imperial, because he's fantastic, has done the statistical field theory side of things, and we're still working on that. And also my students, because, well, I just want to do a bit of advertising here. If there's anyone that wants to visit Chile or even come for a master's or a PhD, just let us know. We are doing things on neurons with Javier and some biologists here in Magdalena. I'm now starting a project on machine learning, reinforcement learning, and how agents interact. I'm also working on developmental biology with Catalina, doing some optics with Tomás Troncoso from another university, even working on bacteria. So there's a wide variety of things that I'm interested in. As long as it's non-equilibrium statistical mechanics, it's fine with me, and it's fun. And of course, my funders. And with that, I'm done. And many thanks. Well, thanks a lot for listening. And if you have any questions, please ask.
Thank you, Ignacio. Thank you very much for your nice talk. We have some questions from YouTube. Let me pass them to you. So we have a first question that is, are these methods suitable for studying vegetable leaves?
Yeah. So there's a whole group of people that study leaf venation. I from the top of my head, I can recall Eleni Katifori. She's a researcher working on leaf venation, and there's some shared mechanisms. We were at a conference. There was a conference a few months ago that combined the communities of leaf venation and river dynamics and also branching morphogenesis. And we were discussing if there were shared mechanisms, and it seems like there is. Especially there's some rules on the duct width. It's called the Murray's law that relates the width of a duct with the width of the two sibling ducts. And you can imagine that should also be valid in organs in human or mouse that transport fluids, right? So yeah, there is a connection. There is a connection. The mechanisms are different, but there
Thank
is a
you.
yeah.
Nice. Another question from YouTube is if it would not be possible to genetically mark the salivary gland cells to show fluorescence.
Yeah, so that's exactly what we did. I didn't show the actual data here, but in that developmental cell paper, what we did was to label individual cells during development. So we labeled an individual cell at embryonic day, let's say 14, quite early on, where the tree has only 100 ducts. And we label at a very low density so that we know that we're labeling an individual cell and not multiple cells. Yeah. And we looked at the tissue 4 or 5 days later. Yeah. So that cell has developed into now multiple cells. That's what we call a clone. Yeah. So a single cell has developed into multiple cells. But those cells are distributed within the tissue. So this single cell that might have been a tip cell has now formed ductal cells, acinar cells, myoepithelial cells, a whole progeny. And based on that, we can start understanding the hierarchy and how the tissue is being formed by single progenitors. So we've done that, and you can do it. We've also activated mutations like cancer, but that's for another day, I guess.
Okay, thank you very much. Let me check if there are other questions in YouTube. No, I think that's all for the moment. But I have saw that you have had show your email address in some slides,
Yeah,
so
yeah,
the
no, no, no.
students can contact you
Yeah, yeah,
later,
please.
because
Okay.
many people do not see the webinar now, but the webinar will be available in the channel of YouTube from the Brazilian Physical Society, so many people can see it in another time, and
Of course.
maybe some student will be interested in your work and can contact you later.
Yeah, of course.
Okay,
Students and even possible collaborations with colleagues in Brazil would be great. We should come together. We're in Latin America and we keep collaborating with people in Europe. We should have a more collaborations locally, I think.
sure, sure, nice. Okay, thank you again, Ignacio, for the wonderful talk.
Thank you,
So
Marcelo.
we can finish this session now, and I invite the community to bring other names for the next webinars. So that's it. Thank you. Good night for everyone. Bye bye, nice. Thank
Good
you
night.
very much.
I see you.
See you.
See you. Bye bye.
Bye bye.